The next thing we're looking at is diffusion. Diffusion is when molecules move from a high to low concentration and they move without any use of any energy. No ATP is burnt. This is the exact opposite of active transport, where it moves from a low to high concentration or against the concentration gradient and uses ATP. And our body will do both. It will diffuse materials into and out of the cells. We use diffusion for breathing, moving oxygen into the cells, but we will also do active transport to move certain materials into and out of the cells also. Now with diffusion, things that will affect this are going to be the size of the molecules, the concentration gradient itself, if there's a large difference between the two sides, whether there's an electrical charge or not, temperature will also affect it, and the size of the molecule itself. So we are also the medium that we're going to diffuse through. So we're going to start with showing diffusion of potassium permanganate in a dish of water. Now water is loosely packed together, so it's going to be easier for the molecules to diffuse through, versus if it's a tightly packed material, then it's going to take longer and be harder to diffuse through. So with this, we're dumping in a potassium permanganate crystal, and we're going to start. It's very dark purple in the center, and you can already see that the water is starting to diffuse and change color. The dye is moving outward, and there is movement from high to low, and given enough time, the whole water will turn the purple color, the lighter purple, because it will diffuse equally throughout it. Now with this medium and with the agar gel medium we're also going to look at, we're going to calculate the rate of diffusion. So with this we would measure how far it moves within a 2 or 3 minute period. And we have underneath the dish, we have a grid paper which is marked in the dark lines or centimeters and millimeters. So we can actually measure this diffusion in 2 to 3 minutes. So I'll give you an example. Say in 2 minutes it moved 20 millimeters. You can then calculate the diffusion rate, or 20 millimeters, the diffusion rate of dividing the 20 millimeters by the 2 minutes, and that would give you millimeters per minute. And diffusion is going to be much quicker in water because the molecules are not packed tightly together. The second diffusion we are going to look at, and we're using the same chemical, but we're going to use 2 different chemicals, and we're going to do it in an agar plate. This is agar. Agar is a thick gelatinous material that we grow our bacteria in for nutrient medium. And we're going to put that paper right underneath this also, so we have a grid to look at. And we have taken 2 little holes, and we have put the blue dye, which is methylene blue, which has a molecular weight of about 320. And we have put potassium permanganate, the golden one, in the other well, and that has a molecular weight about 156. So potassium permanganate is a much smaller molecule than the methylene blue. We've started them at the same exact time, and we have measured the diffusion in 15-minute intervals over a 60-minute period. And just visually, you can see that the potassium permanganate has moved much farther than the methylene blue during that time period. You want to measure it from the edge of the well out. So you can see it's moved about 10 millimeters or so, whereas the methylene blue has maybe gone about 3 millimeters during that same time period. We did it over a 60-minute period or an hour period. We could divide this by 60 minutes to get the diffusion rate per minute, or we could divide it by how many hours if we ran it for 2 hours to get diffusion rate per hour. Comparing the diffusion rate in the gel to the diffusion rate in water, water diffuses much quicker, and molecules diffuse much quicker in water. And comparing the size of the molecules, the larger the molecule, the slower the diffusion rate. The smaller the molecule, the faster the diffusion rate. Okay. The only other thing that you are responsible for on this lab, and we are not going to provide any visuals for this, but you will look at your book and pictures, is what happens to red blood cells when put into a hypertonic or isotonic solution. Now, with your red blood cells, they have the plasma membrane. They will lose fluid in a hypotonic solution. Gain fluid in a hypotonic solution, so they will swell. And if pushed far enough, they will actually rupture, and this process is called hemolysis. With your red blood cell in isotonic, it will stay the same, nice bioconcave cell. And if you put that in a hypertonic solution, it will lose water because it will diffuse from the high inside the cell to outside and will shrivel. And this is called crenation. Okay, look at those pictures in your lab book because there are pictures of the red blood cell, but we are not setting up the slides for that.